Hello viewers, I will discuss about the advantages and application of mutation. The mutation may be favorable or unfavorable selected by the environment where the system is present. The advantages in general include the evolution, natural selection, the survival strategy, genetic variability, diversity or polymorphism, adaptation as well as repair system. Let's discuss about them individually. First one, evolution. As we know that according to neo-Darwinism, the species are getting evolved by evolutionary mechanism where there are several reasons. Mainly two reasons are there, and one of the best selected reason is the mutation. So, mutation help the evolution to go a proper pathway. Natural selection. As we know that in a particular gene pool, there are frequency of different allele. From the wild type, we may follow the variation of the allele, but the fitness of the individual is selected according to the environment present. If it is positively selected, so that can be delivered to the next generation. So, we know the survival is dependent upon the fitness. Let's take the example of the Biston betularia. As we can find, these sort of moth have light color as well as dark color phenotype by mutation. But light color phenotype can escape very easily, but dark color cannot. So, dark color can be destroyed by predators. Genetic variability. So, by mutation, different kinds of genetic alleles are introduced within a particular gene pool, and it gives different allele for genetic testing, genetic analysis as well as genetic structure of population, because in case of different kind of genetic analysis like Mendel 2 hybrid cross, different types of mutant alleles are given which is taken for the testing. Diversity. So, in this way, the physiological, the behavioral as well as biochemical changes can be happen with respect to a particular loci which is getting mutated, giving different variable allele, and these phenomena of at particular loci having different types of mutant allele which is getting manifested at the phenotypic level is called polymorphism. So, better polymorphic thing by the natural selection, by mutation is getting selected. Repair system. According to the mutation present, the repair system is getting evolved from the earlier one. As we know that according to them, if repair system missed any sort of mutation, so that may be introduced to the next generation. If it has gene into environment positive interaction, so it is called beneficial mutation. If it does not, that causes harmful mutation. Usually harmful mutation are obsoleted from the gene pool. They are naturally selected. Adaptation. So, as we know that survival of the fittest by natural selection, when the beneficial mutation is getting accumulated, so that gives the better species to evolve, and that is after accumulation by reproductive success is transmitted to the new generation. Like butterfly, according to the predator-prey relationship, we know that if the phenotype that is mutated is better from that of wild type which can escape from the predator, so that will be better evolved, naturally selected, and more number of the next generation population can be created. So, in this way species can be evolved, and in this way we can find the adaptation. So, we are talking about the beneficial mutation, but beneficial mutation follow some of the characters. The character includes that the traits that we are going to discuss, that is phenotypic expression, may be favorable or unfavorable depend upon the circumstance where the organism belongs to or the environment. Number 2 criteria is that on what extent the gene is fixed, whether the mutation is stable or not on that particular environment, it is to be noted. And the third point is that the genes responsible for the follow-up of the trait must be taken account, because some polygenic traits are there, okay, some oligogens are responsible to show some beneficial trait, so that will be taken account. Let's talk about the beneficial mutation with respect to physiological aspects. So beneficial mutation can be observed in case of human against some of the disease. So disease resistance is found by the mutation. As well as in bacteria, we will find the antibiotic resistance as well as the bioremediation. And in case of plant, by mutation, it is induced to have the desirable traits for crop improvement. Okay, next one. Let's talk about the disease resistance in human by mutation. First one, ovarian cancer. It was found in some of the women having BRCA gene mutation, giving short time survival, because from 2 years they can survive more 3.5 years having mutation in BRCA1 as well as BRCA2 genes. So by which ovarian cancer patient can survive. Colon cancer, MTHFR 5,10 methyl tetrahydrofolate reductase converts 5,10 methynyl tetrahydrofolate to 5-methyl tetrahydrofolate, which is very responsible for the homocysteine to degrade and to produce S-adenosyl methionine, very important for the DNA in vitro or in vivo methylation. If this gene is mutated at the 677 position C to T or at 1298 position from A to C, they are resistant against colon cancer and in case of some of the leukemia. Next, Alzheimer's disease. As we know that in case of neurodegenerative, this sort of disease that was studied in case of some of the Icelanders, it was found up to 85 years they become resistant against Alzheimer, because there is a protein called the amyloid precursor protein APP, which is disintegrated by secretase enzyme, gives amyloid beta, which gives the toxic protein to be accumulated. If APP become mutated to be attacked by secretase, they do not produce amyloid beta, so person becomes resistant against Alzheimer up to 85 years. We can find the lactose tolerance, because after weaning period, the lactase gene become switched off by mutation. Then again it can be switched on in the dairy consumer population by mutation. So in this way lactase mutation give rise to the lactose tolerance. Against the Larsen syndrome at FLNB gene, which is responsible for filamin B protein to be synthesized, the mutation gives against the diabetes resistance. Malaria resistance. As we know in sub-Saharan African population, if at globin chain at 6th position A is getting mutated to T, so from glutamic acid it is changed to valine, give truncated hemoglobin formation. So person will suffer from sickle cell anemia. But we can find in case of sickle cell trait heterozygous condition, the person will be escaped from malaria because plasmodium cannot attack sickle cell RBC. Reduced heart attack. In case of atherosclerosis, as we know, apolipoprotein AI can remove the arterial plaque by cholesterol removal to the liver. Now, in case of Milan, it was found apolipoprotein AIM, which is much more mutated and responsible to remove arterial plaque better. So, it helps to fight against atherosclerosis. In case of PCSK9 gene mutation, also we can found against heart disease, the risk is getting reduced. In case of some of the bone density increase, that was found in case of some Midwest accidental case by mutation at lipoprotein related receptor 5 give increased bone density, which actually helped them against the cancer. It was found by the X-ray. Next, the mutation in case of tetrachromate vision. Over the X chromosome, there are some of the cone gene that helps in case of recognition of red and green color. If they are mutated in case of some of the women, so tetrachromate vision can be observed, the extra gaining. Next, very important, if the chemokine receptor 5 is getting mutated, so HIV cannot attack on that particular chemokine receptor, because as we know that HIV attack to some of the macrophages over that receptor. So in this way, on a particular HIV susceptible population, they become resistant against HIV by this sort of mutation. In case of bacteria, we can find the antibiotic resistance as well as microbial biodegradation. Antibiotic resistance, as we know that by the presence of antibiotic, the microorganism become resistant. Like quinolone resistance was found by guide A and guide B gene, as well as the beta-lactam resistance was found by PBP2X mutation. Microbial biodegradation or bioremediation was found by in case of nylon-eating bacteria and oil-degrading bacteria. In case of nylon-eating bacteria, if nylon is present, by some of the shifting of the frame, new enzyme is getting evolved to degrade such biopolymer. And in case of oil-degrading pseudomonas also, we can found the novel enzyme. By this sort of mutation, the mutated enzyme is able to degrade the hydrocarbon. Next, beneficial mutation to crop improvement. They help in case of mutation breeding and germplasm screening. Let's talk about the mutation breeding, that is induced high frequency mutation is given to some of the plant to increase the polyploidy, which actually help to give the desirable trait like hull-less seed, protein content higher in case of wheat and maize, etc. As well as it actually helps in case of other purposes for the instant speciation, because the polyploidy of interspecies can evolve better evolved plant species. Germplasm screening, as we know that mutation help to have polymorphic allele by which the population can be studied by germplasm screening by marker assisted selection. Next one. So, use of mutation in case of biochemistry actually help to know about the biochemical pathway. Number 1, mutation defines biological function. Suppose E. coli is able to uptake lactose at 10 to the power minus 5 molar. If they are unable, so there must be some genetic makeup for that particular lactose uptake. So in this way, it helps to give rise to the idea of lactose transporter. So it helps to know the mutant type about the biological functioning. It helps to elucidate biochemical pathway by mutational block. Let's talk about the galactose metabolism. So in case of galactose metabolism, we have gal E minus, gal T minus, gal K minus mutant. In what is going on by experiment? The radioactive 14C galactose is given and the accumulation of the metabolites were observed. It was first found in case of gal K minus mutant, the accumulation of 14C radioactive galactose. But in case of gal T minus mutant, it was found the accumulation of galactose 1-phosphate, which is radio-labeled. But in case of gal E minus mutant, we can find not only 14C galactose 1-phosphate, but also UDP galactose is getting accumulated. In this way, that actually helped to elucidate the total pathway. When the 14C galactose is accumulated, so galactose kinase is mutated, which is responsible to convert galactose to galactose 1-phosphate. And galactose T product, gal T minus product, gene product is responsible for the conversion of galactose 1-phosphate to UDP gal, which is getting converted by gal E gene product or epimerase. So in this way, total biochemical pathway can be elucidated. Not only that, biochemical regulation can be formatted. In case of galactose metabolism, all these mutant products are found in gal plus, that is non-mutated condition, which is able to utilize galactose. It helps to know the interrelation between biological and biochemical interrelated functions. Suppose in case of Pol A minus mutant, it was found DNA polymerase capacity is 50-fold is getting reduced. So, polymerase 1 is not the sole thing for the DNA polymerization, because backup polymerase 2 and 3 are there. Actually, polymerase 1 is responsible for the repair also. So, in this way, from Pol A mutant, we can know there are some other polymerase responsible. Mutation help to correlate the function of two unrelated systems. Suppose if E. coli is unable to adsorb lambda phage, they are also unable to absorb or to uptake maltose. So, maltose uptake as well as the lambda phage absorption is somehow related. Mutants actually help to know how two proteins interact. In case of lambda phage, it actually helps us to know that lambda replication needs the P protein and DNA B protein to interact with each other. And it actually helps to know how biological agent interacts with the external agent. Like in case of rifampicin sensitive microorganism, the RNA polymerization is inhibited. In all the cases, the biochemical mutants help to know the high resolution dissection of biochemical or biological processes. It actually includes the metabolic pathway, it includes the morphogenesis, it includes the photosynthetic electron transport chain in Chlamydomonas reinhardti, it includes to know about the development and behavior and some of the characters in plant, animal, including the Drosophila. Application of this beneficial mutation is done in case of plant for having desirable trait by crop improvement by mutation breeding, and the beneficial mutation, that is antibiotic resistance, was utilized by some of the drugs by bacteria. Okay, so that actually helped them for the positive screening, for the positive selection. Next, geneticists assay the mutants for mutation analysis for the correlation of biological and biochemical function. And these mutation helps to have higher yielding antibiotic, because the first generation antibiotic synthesizing penicillium, if the spores are irradiated, they give higher yield of penicillin, and genetic screens serve better phenotype. Mutation which are beneficial is needed for the cancer therapy as well. Next one, mutation breeding. So, mutation breeding is the most of the application of beneficial mutation. That is, the desirable trait is introduced in the plant for the crop improvement having this objective. So, apart from objective, let's talk about the process, application, limitation and achievements. The process involved to select the part of plant, maybe seed, maybe pollen. The dosage, according to the system, the dosage can be vary. Irradiation treatment, maybe if it is irradiation, then just after irradiation it will be planted. If it is chemical mutagen, then it will be pre-soaked, and dosage will vary according to the variety. Next, frequency, then recurrent irradiation must be given, and after that it will be planted for M1 to M8 generation. So, gamma garden is a confined area where gamma radiation is given to evoke the polyploidy. So, actually it helps to save from the animal attack and help in case of direct mutagenesis. So, polyploidies actually give rise to the interspecific speciations, okay, and they actually give rise to spores. That means the part of the plant having polyploid in number, having desirable trait. By aneuploidy also, sometimes the desirable trait can be manifested in the plant. Limitation. So, apart from all these advantages, mutation breeding has limitation like low frequency and very higher population must be assayed, okay, and not only that, interspecific translocation may sometimes inactivate some beneficial genes also, and we can have most of the mutation is recessive. So, from the disadvantageous mutation, we have to take up the beneficial mutation having desirable trait by this sort of mutation breeding. And mutation breeding is actually applied to maize, to barley having hull-less seed, stiff-less straw, higher protein content, etc. Let's see what are the achievements. In case of wheat, NP836. In case of jute, JRO514. In case of cotton, Indo-2 cotton is very important. And sugarcane, CO8153. They are all the mutated gene product coming from the polyploidy, which are giving for the instant speciation having desirable trait. So, as conclusion, the beneficial mutation is needed for the desirable trait introduction for crop improvement, resistance against the disease in human, the antibiotic resistance in bacteria help them for positive screening, and in case of biochemistry, they help to elucidate the biochemical pathway. And the morphogenesis was found in case of the T4 lambda fudge, and which actually helped to introduce the three-dimensional structure of protein after each case, which helped to modify the total morphogenesis process, which were isolated by temperature sensitive mutant and observed by the electron microscope. Thank you.